All right, good morning, everyone. I'd like to welcome you to this session in Early and Minimal Life. My name is Alonso Ricardo, and I'm going to be chairing this session today. I also want to thank the organizers for inviting me and giving me the opportunity of being here today. We have a very exciting set of speakers this morning in which we're going to try to start seeing those first glimpses of what we call life. Just to give you a little overview of the kind of things that we're going to see, I thought that I should give you an idea of where are we starting, basically. What is the beginning of today's talks? So it's a little different. So tomorrow we're going to be talking about prebiotic chemistry, but today we're making the assumption that prebiotic chemistry had already happened, or basically that some of those building blocks that are going to be used for the construction of what we call the early and minimal life were already available. So basically, there are two sets of you may call strategies to try to address this problem. And one of them is basically to borrow components from modern life and try to use those components that we know exist today and just try to use modifications of them to understand how that early and minimal life could have started. So as you can see, today you're going to see some of the talks that basically are using nucleotides or activated versions of these nucleotides. Some of them using deoxynucleotides, and this is a very interesting molecule which was presented by Matt Towner on Monday, and basically it's also an activated form of a nucleotide. And we start from the assumption that there was already some sort of RNA or DNA. Whether it was able to self-replicate is just there, but that's the transition that we're trying to address today. We are going to see a lot of membrane components as well, mainly phospholipids, but also the lesser part of the phospholipid, which is basically the fatty acid component, that are able to self-assemble and make these structures that we call vesicles by themselves. We'll see also some more of the later part of these amino acids components in the peptide and protein work. The next part, and we're going to see some of this, is what is the other trio of approach, basically, in which we're not borrowing ideas from modern life. We're actually just a little, but we're trying to basically just design something. So you don't worry that much about the abundance of the building block. You just kind of design your own building block to perform that specific function that you want it to have. So you can see some of this on the poster sections, but also you're going to see some of this during one of the talks. And just to give you an overview of how people are approaching this, or in particular how I look at it, and some other people on working on this field, is this idea of basically modifying the nucleotide. So as you can see, this looks like a nucleotide. Basically, this is cytidine, which has been modified, and a group has been attached here, and the nucleophile has changed. And these changes allow this molecule to basically start polymerizing without the need of any protein enzymes. You can get more exotic. You could go and change your sugar and make it look now like a morpholino ring, which is represented in here. And all these molecules, and if you want to get a little more extreme, you can change it for trios. And all these molecules have different properties, basically. They will adopt different geometries when they are making duplexes. And perhaps they will tell us a little about how this replication process actually arose on the planet using the normal building blocks. You can see some of the posters here that had some of this information. Also, the other thing that people working in synthetic biology, or before actually life appears, we'll call it synthetic chemistry, is components that are basically mixtures, and trying to see how this mixture of components will give kind of a selected advantage to a particular protocell. And so there is a poster by Itai Burin in which basically he's just presenting just that. So how the inclusion of phospholipids into vesicles will give an advantage to the vesicles that contain this material over the other ones. For the amino acids, I think people had approached this in a different way, and most of the work with amino acids had been done towards constructing proteins with selective modifications so that we can put other things in there. But you know, we can always start looking at things like D-amino acids, for example, which since we don't have the restriction of working with a biological system, we don't need enzymes, so we could definitely start looking into something like that. And there are other group of people that basically work with hybrid-like molecules, which are basically mixtures of of some of these things in here. And you see this on some of the work of PNA. You see this in some of the work of nucleotides, basically, which are activated with lipids. And some of the work by Pierre that's going to be presented today with some of these lipid nucleobase complex. So just the last slide on my work, basically, is just the status of where we are. And this is the particular case for these aminonucleotides. So basically what we did was to construct all these different nucleobases that, as I said, had the ability of basically organizing themselves and be reactive with each other. For example, if this amino group will attack this phosphate, and basically you will have now a phosphoramidate bond, which is different from a phosphodiester bond that is regularly found on DNA and RNA. But they form, and they form in the same way that you will expect an enzyme to do it. So if you take a primer, basically, which is just a small piece of DNA that is aligned into another longer piece via Watson-Crick complementary base pairs, and you just add this with no enzyme and anything else added that will affect the reactivity, what you'd see is that they basically just start aligning in front of the template, and then they just form chemical bonds. The process, we're still trying to figure it out if it is a process that basically happens one by one, or if perhaps there is pre-organization so that this template is populated by a lot of these little nucleotides, and then just the chemical steps occur rapidly. That would be dependent on the temperature, for example, in which you run your reaction. Or perhaps that if these monomers are condensing each other initially into dimers of mixed composition, and then these dimers are aligning in the template and then reacting with each other. But nonetheless, so this is just a quick gel that shows that if you put these 4 nucleotides here, you will finish up this extension and you will see a full length of product in a matter of a few hours. This doesn't come with problems. This comes with some problems, however, and the reason why these problems arise is because you don't have a protein anymore that will be doing your basically proofreading. You don't have a protein enzyme that is going to be correcting mistakes. So you had the opportunity to make errors. So for example, if I will remove diaminopurine, which is this molecule here, we just don't add it to the solution, what happens is that uridine, which will be the propionyl uridine, which will be the molecule base pairing to it, could form a wobble base pairs with guanosine. And that's what happens here. So if we start the reaction, it kind of pauses there, but then slowly guanosine starts making this wobble base pair, and then basically the thing can finish up. So that's basically the problem that we had, and it's all regarding to this wobble base pairs with propionyl uridine and guanosine. But there is a potential solution to this fidelity problem, and you can look at it in the poster of Bain-Helberger in P554. All right, so now going back to the discussion today, the way I see this is a definition, but again, and it's synthetic life. And what do you call minimal? So there are 2 approaches that are considered to be relevant to this discussion, and are those that are basically bottom-up, and are those that basically try to just basically design and reconstruct an artificial system that looks kind of like this. Basically has a compartment, it has a self-replicating informational polymer, and that has the ability to, obviously, to evolve. And it has an external energy source, so the food's coming already from outside, and it already has the energy necessary for this replication process to occur. And then there is the top-down approaches, in which basically, this is more biology. Basically, we take an organism and start stripping out all the genes that we think that are not important, and you get to things like this that Craig Penter has worked with, which are basically containing membrane, DNA, genome, RNA, proteins, and metabolism. So now this is a figure that I borrowed from a paper that we published in Scientific and American, and basically this is kind of the way the discussion is going to look like today. So we're going to start from the assumption that there was some RNA, there was some polymer, and that this polymer was basically making copies of itself, or basically this is not a replicase just yet, this is just basically making a complement by using activated nucleotides that are entering from outside with this elemental compartment, which could be a fatty acid containing vesicle. Now this nucleic acid will, by convective flows, basically just heat and cold sources, could move around, and basically this starting what we call here protocell could start dividing, and then the strands will separate via thermal heating, and then just by adding more fatty acid molecules, this protocell could divide, and then you could have 2 daughter cells with information on it. Some open questions in this particular slide, for example, what type of monomers would you be using if if you're in a primordial soup? Were they actually monomers or they were short oligomers? We don't know that much about. I guess Irene will probably discuss a little bit about the fidelity of this process. So how do you deal with errors on a system where there is not an enzyme? And also, we don't know that much about the chemical and perhaps mineral catalysis present at this period. Now we move to a second part in which now we assume that there was already abundant species of RNA that were encapsulated into a lipid membrane. Some of these RNA molecules will learn new tricks, and they will start folding into molecules that will allow to improve its own reproduction, and at some point it will allow the appearance of what we call a replicase ribozyme, basically, that will allow the copying of one of these RNA strands. And then some other ribozymes will start evolving, and then you will get things that will probably give advantage to this vesicle containing this molecule over other vesicles. You could learn new things, like you start learning to use nutrients from outside by a complete set of ribozymes. We're going to see some of this here as well. We're going to see some of the ideas in trying to modulate this complex networks of ribozymes. Again, some open questions here. This system is supposed to be self-sustaining. It's coupled to membrane chemical evolution, and some people call it metabolism. I call it complex chemistry, in which building blocks are produced and are being used actively by some of these ribozymes in here. So these proteins now will start taking over the catalytic functions as we see in modern biology, and now the energy processes and all these things are perhaps going to be done by proteins using things like proton pumps or sodium pumps or things like that. However, there's also going to be perhaps a strong selective pressure where proteins are going to start doing the transport of nutrients in and out the protocell. And at last, we're going to see some of the work in between, this transition between DNA and RNA. And it will be interesting to see if perhaps this transition to DNA to RNA actually occurred this late in the process, or perhaps was much earlier, that they were both coexisting at some point. We don't really have a good prebiotic synthesis scheme for the synthesis of the oxynucleotides, but that doesn't mean that it doesn't exist. Some of the intermediates, for example, in Matt Pauner's and John Sutherland's work, could serve as starting points for thinking about sulfur chemistry that could be used to form the oxynucleotides on an earlier. We also need to learn about the origins of amino acylation and translation, and the role of genetically encoded proteins, and in particular the evolution of these genetically encoded proteins, which Andrew is going to tell us a little about how that process happened. So without further delay, these are basically the speakers this morning, and I would like to invite Irene to come over here and start telling us about the first part, which is basically information-rich sequences from replicating systems. And also going to ask all the speakers to keep to their time. We have a lot of talks today, as you can see in this very session in the morning. So thank you very much, and I hope you enjoyed this talk this morning.